Welcome to the AI Research Roundup. I'm Alex. Today we are looking at a paper from the Hugging Face trending list published on September 29th, 2025, which was about 6 days ago. The big hook is that this paper introduces LLM DNA, a compact, task-agnostic fingerprint of a model's behavior, and shows strong relation detection with an AUC of 0.957. The paper is titled LLM DNA, Tracing Model Evolution via Functional Representations. And as we will see later, the implications for model lineage and management are quite significant. Beyond relation detection, the study delivers a training-free extraction pipeline, competitive routing, stability across prompts, and evolutionary trees that mirror real architectural and temporal shifts. Alright, the introduction laid out the motivation and the two guarantees. Figure 1 shows the training-free extraction workflow, moving from a small prompt set to a compact DNA vector. The pipeline collects responses, converts each into a sentence embedding so semantically close answers align, then concatenates them, and applies a random Gaussian projection to about 128 dimensions. The design remains model agnostic and works with closed-source APIs because it relies only on outputs. Well, Figure 1 outlined the extraction pipeline. Figure 2 shows the DNA distribution for 8 models relative to Llama 2.7 BHF projected into 2 components. The blue points mark independent models, and the orange points mark correlated ones, and an SVM with an RBF kernel draws the dashed decision boundary. The two groups separate cleanly, which signals that the compact DNA vectors preserve meaningful functional relationships. So the previous figure showed clean separation for relation detection. Table 2 presents routing accuracy, which measures how often the router sends a query to a model that answers correctly. Random hits about 0.40, while single best reaches about 0.61. Embed LLM with matrix factorization scores about 0.67. DNA edges it out at about 0.67 as well, slightly higher, which is notable because the DNA vectors are frozen and extracted without training on the routing task. Alright, the previous table compared routing accuracy. Table 1 reports relation detection across 305 models, listing precision, recall, F1 score, and AUC. DNA reaches about 0.86 precision and about 0.92 recall, with an F1 near 0.88 and an AUC around 0.96, which clearly surpasses random and greedy. The higher recall than precision suggests many flag pairs are real, because some relationships likely remain undocumented in the official model tree. So the previous table reported relation detection metrics. Figure 5 presents a phylogenetic tree built from DNAL2 distances using the neighbor joining method, which organizes model groups by functional similarity. The branches trace a shift from encoder-decoder families toward decoder-only lines and track a rough timeline from 2023 to 2025. Longer branches for Qin and Gemma suggest faster functional change than the Llama family. Alright, 2 takeaways stand out. LLM DNA gives a compact, training-free fingerprint that preserves functional distances, which explains the strong relation detection around 96% AUC and competitive routing near 0.67 accuracy. Broader impact is practical provenance tracing and phylogenetic maps that reveal real architectural and temporal shifts. That is it for this episode of the AI Research Roundup. I am Alex. Thanks for listening.